Technology principles and process. Restriction endonuclease. This is very important for MCQ. This is an enzyme which recognizes a specific palindromic nucleotide sequence in the DNA. It cuts the DNA molecule at identified position within the DNA. This enzyme cuts the sugar phosphate backbone at specific sites on each strand of double helix. That is, it cuts the both strands of double helix. What is the meaning of palindromic nucleotide sequence? Palindromic nucleotide sequence in the DNA molecule, they are groups of bases that form the same sequence when read both forward and backward. Restriction endonuclease is identified from E. coli in 1963. First restriction endonuclease is HIN2, isolated from Haemophilus influenzae (RD). Restriction enzymes belong to class of nucleases. There are two types of nucleases, exonucleases and endonucleases. Exonucleases are enzymes which removes the nucleotides from the ends of DNA. Endonucleases, they make cuts at the specific positions within the DNA. This is used to remove and this is used to cut. Restriction endonucleases used in genetic engineering are used to form recombinant molecules of DNA. EcoR5 is the type 2 restriction endonuclease. It is isolated from E. coli which produces blunt ends. This is important MCQ. Restriction enzymes which produces blunt ends and which produces sticky ends. Restriction enzymes which produces sticky ends are EcoR1, BMH1, HHA1, XHO1, HIN3. Restriction endonucleases EcoR1. E stands for the first letter of genus name of the bacterium. Coli stands for first two letters of its species. And R is the strain of the organism. The last Roman numerical 1 is the order of discovery. Restriction endonucleases, they are synthesized by the bacteria. Restriction endonucleases are enzymes synthesized by the bacteria as a part of the defense mechanism.